Good afternoon and welcome to an EAS Consulting Group webinar on cleaning validation presented by EAS independent consultant Joe McGinnis. EAS, a member of the certified group of companies, is a global leader in regulatory solutions for industries regulated by FDA. Our global network of over 150 independent advisors and consultants enables EAS to provide comprehensive training, auditing, and consulting services, ensuring proactive regulatory compliance. From strategic product development and preparation of technical submissions to assistance with Form 483s or warning letters, EAS offers the detailed knowledge and experience your company requires. EAS consultant Joe McGinnis utilizes extensive experience in the pharmaceutical industry to assist clients with preparation for and execution of both internal and FDA mock audits. He designs protocols for SOPs, GMPs, and training programs, ensuring an understanding of regulatory compliance requirements, and assists with remediations and responses of CAPA findings and deviations. He also helps with the preparation of DMFs in support of FDA submissions. During the webinar, you may ask any questions by typing them into the questions box, and we will save all questions for the end. And with that, I will say thank you for joining us, and we'll turn the presentation over to Joe.
Okay, thank you very much. Today's topic is cleaning validation. Your GMPs depend on it. I hope everybody can see my my slides. I'll tell you that my main area of expertise throughout my time in industry was with active pharmaceutical ingredients. So I might bring a somewhat different perspective to this if I trail off too far into APIs and I'm not talking enough about your application. Feel free to ask questions about that, but I think we should be okay on that. Okay, most of these slides are going to have a little picture on them. I think that helps break the thing up a little bit. Some of them will be amusing, some of them won't. Some of them that I intend to be amusing, you might not find very amusing. You know, cleaning validation, you would think, well, that must be a mature topic. That must be something everybody does now. Nobody really worries about anymore. But that's not really the case. I mean, we had a warning letter just issued in the last few weeks by the FDA to a firm, and it said things that you could have read, and in fact, we will read about in literature that was published 30 and even 40 years ago. This is almost boilerplate language. Your cleaning procedures for non-dedicated equipment are inadequate. Our investigator observed drug residue inside one of your tablet presses, documented clean by your quality unit. And I will tell you that in another life, I've seen something like this almost identical in the last year or so. The firm tested the residue, as you would want to do during your investigation, and they found drug substances from previous processes, and that was plural processes. So residues have been hanging around in that equipment for quite a while. And you get something like that, you're going to look like this poor fella here. He looks a lot like me. I used to have hair like him too. But what this has happened to this firm is they've put all their product in jeopardy, right? They've by not dotting their I's and crossing their T's as they go from product to product in a multi-product plant, they have endangered consumers, and they've endangered their own license to operate. So in the warning letter, the FDA asks for them to test all reserve samples that were manufactured on your tablet press, and that's going to be everything within expiry. And they're going to have to test those samples for everything that they made in there, which means there's huge amounts of analytical development that is in front of this firm. They want an assessment of all potential sources of contamination, not surprising. And they want a comprehensive, independent, so read, you know, again, cost, retrospective assessment of your cleaning effective to evaluate the scope of cross-contamination hazards. One of the things that bothers the FDA by far the most is the notion of cross-contamination, of a person perhaps allergic to a substance is being exposed to something that they don't even know is in the drug itself. And then, of course, a CAPA plan that includes remediation to processes and practices. And I'm sure FDA is going to be worrying about the R word, recall, may in fact be in there. And Captain Picard is not letting me. Oh, there we go. Okay, let's make sure. Okay, good. The warning letter cites the FDA's worst case list, and that's my characterization. They've been talking about worst case for a really long time. Because if you sit down and you think about it, if I'm going to validate my cleaning procedures in a plant where I might make, let's make it simple, 10 products, well, think of the number of possible combinations of made a product in a piece of equipment, what's coming along next. There's, many, many different possible combinations there. And so what they're going to want you to do, I mean, you certainly could try to validate every possible scenario, but eventually you would have to retire and somebody else would have to pick that up. I mean, it would take you forever, in other words. You don't want to do that. Cost prohibitive, et cetera. So FDA says, let's talk about worst cases. So they talk about you have to consider drugs with high toxicity, drugs with high potency, drugs of lower solubility. But it's not just drugs, right? Obviously, it's ingredients of lower solubility, right? We tend to think of the active ingredient as being the most important and the most potent thing in a drug process. Let's say it's an oral solid dosage form and we're going to do a formulation. Well, I got rid of the active ingredient. Isn't that wonderful? My tablet press is clean now. Hold on there. What excipients did you have in the process? And did you clean those as well? And people can have allergies to those as well. So it's not just, when you say drug, we mean, ingredients. They want you to worry about swabbing locations for difficult to clean areas. How do you define that? What justification do you have of that? Residues for use in recovery studies. So what that means is how hard is it for me to detect a drug residue on the equipment that I'm operating on. The famous dirty hold times. Clean hold times is something that you used to hear about 20 or 30 years ago, and you still hear about it. But more recently, you hear about dirty hold times because usually when you clean your equipment, almost all firms are going to put it away put it away, quote unquote, in a way that it doesn't really get exposed to the environment, right? So after a while, is it going to get dirty again? Odds are no. But if I just let it sit around and I don't clean it for a while, is something going to bake on there and become very difficult to clean? Well, just ask anyone who did the dishes for dinner last night, right? If they waited a while, maybe something got really hard on the frying pan and it was difficult to clean. Okay, we'll talk a little bit about the history of cleaning validation, and then we'll go into, because I think it's very interesting, and I think it illuminates why cleaning validation gets done. And then we'll talk about, some of the modern concepts. How do you set limits and those sorts of things? Okay. A good summary. And by the way, at some point you will you'll receive through EAS a list of resources. I didn't put together and should have a list of good resources that you can click on, so that you can get some the actual regulations, if there are any, involved in cleaning validation, and articles in the literature and such that talk about various concepts that are important to cleaning validation. And I'll say FDA an awful lot during this because they're the agency I have worked with the most over the years. But the European Union and the FDA often sort of have a race where one gets ahead of the other and then the other one gets ahead, as they formalize and learn more about the concept of cleaning vessels. And right now, I think the FDA is playing a little bit of catch up with the European Union in terms of more formalized regulation around cleaning validation. But I don't think that just because the FDA doesn't have something in writing that they won't try to get the C in current good manufacturing practices, right? Get that current going, right? It's impossible for a regulatory agency to have black and white GMP about every possible scenario. So it's a good summary, as I said, in pharmaceutical engineering, beginning on page 74 in the July-August 2011 issue. And I think if you Google that, you should have no trouble with that. Walsh notes that the first recorded use of practical, achievable, and verifiable was in a Pharm Tech article from 1984, which may be the first in literature that people will read, organized discussion of cleaning validation. Before that, the idea of validating cleaning processes, if you looked in the old GMPs for FDA regulated materials, you wouldn't find that word. You wouldn't find cleaning validation. You wouldn't find that term. Very soon after this, though, the idea that you should clean your vessels to a practical, achievable, and verifiable limit became very important. Now, we'll look at this picture as I have here for a second. That's a person swabbing a vessel. We'll talk about that in a few minutes. And note how carefully that person is swabbing the vessel, and they're making sure that they swab the exact size of the amount and area that they are supposed to swab. And I'm sure that in no one's plant ever are the operators any less careful than that. Okay. Well, cleaning validation as a big deal, right, becomes a topic. Doesn't regulatory stuff always start with some sort of scandal? Didn't the GMPs get their start with elixir sulfanilamide in the 30s? Didn't the thalidomide in the late 50s, early 60s, didn't that lead to a lot of changes in pharmaceutical regulation worldwide? And the answer is yes. So, well, what was the scandal that really pushed everybody down the road to we better be validating our cleaning procedures? And oh, by the way, this picture is from a scandal in the 1860s where a maker of candy in England was adding some poisonous substances because they were cheaper to the candy. And I'm qualifying this by saying we'll start with a scandal because that's where the strict enforcement of the idea of validating your cleaning procedures probably got its start, with a scandal. I mean, it was always important to know your vessels were clean. If you look on YouTube, I would highly recommend, if you can find it, I think it's called No Margin for Error. It was a video made by the Food and Drug Administration in the early 1960s, still has a lot of relevance today. And there, of course, they're worried about cross-contamination, so they put a screen in between two mixers. You know, oh, there we go. We've stopped all the cross-contamination. But the concept's there. We're trying to keep one thing out of another thing. So in 1988, there was a cholesterol drug called Questran, and maybe it's still out there. I don't know. It's a cholestyramine resin, USP, and that's used in that drug. Agricultural pesticides, pesticide residues were found in the drug. How that happened, I'm not absolutely sure how they found it. But the history was, and again, this is in 1988, it began, a contaminated product was made by a division of a large American company, okay? Now, even back then, you know, pharmaceutical supply chains were pretty diffuse. The API was made in Italy by a different American company who had a plant in Italy, and they also, at the broad campus, made pesticide intermediates. Recovered solvents were being used at all of these facilities. Well, not the formulator, but at the all the chemical facilities where the pesticides were made and where the API was made, and that's not uncommon. And they were stored in drums. That's also not uncommon. But the firm making the API was not cleaning the drums in between uses. And I would say today, you wouldn't even want to try to clean in between a non-pharmaceutical and a pharmaceutical use for something like that. You would want to make sure your drums are designated for what they're going to be used for. But you know, in theory, you can do this, right? You could put together a cleaning procedure, and that's how the FDA realized that they could sanction the firm, because they could say that you didn't validate cleaning in between the use of the drums for the pesticide and the use of the drums for solvents that you were going to recycle for the active pharmaceutical ingredient. Now, I'm just going to, an interesting point, the American formulator fought the recall of these drugs in the press. And the reason they fought them is because no one disputed that there was very little danger to anyone based on this. But what there was the potential for danger. In the pharma industry, we sometimes miss something that the airlines do very well, which is they worry about near misses. And sometimes we say, well, we got away with that. That wasn't a big problem. We're learning more and more, I think, to concern ourselves with near misses. And this would have been considered a near miss. So again, the timeline of this is also very interesting. The problem was found by the API manufacturer in December 1987. And they noodled it for a bit. And then they said, gee whiz, we better tell our customer. So they did that 2 months later in February 1988. The customer said, well, they noodled for a bit. And then they said, well, we better make sure we're not killing anybody. So they went out and they informed the FDA, but they informed the FDA a month later, after they had got their toxicologists and got together and said, well, you know, these levels are really small and it's not going to be a real problem. Then the agency and the company sat down and they noodled for a while. And it wasn't until mid-May that a recall was announced, mid-May of 1988, that a recall was announced for this product. Now, just letting you know, today these delays might bring regulatory sanctions. They could bring criminal prosecution to the extent that FDA was involved in any of the delays. Well, congressmen like to get on TV, and so they get on TV and they make the FDA, you know, sit there and sweat. And that's how the FDA gets kept on the straight and narrow. And it will be the same in Europe and other countries around the world where, you know, we want to keep the regulatory agencies on their toes. So, you know, things have changed over the last number of years. So this brought a famous FDA investigator to the forefront, and I couldn't find a newer picture of him than his college graduation picture. And his name was Hank Avalon. And Hank Avalon blew through especially the active ingredient industry, but really through the Northeast United States for formulators, oral solid dosage form, parenteral forms, like a whirlwind in the late 1980s and early 1990s. He really had the idea that we need a scientific, organized basis for what we are going to ask firms to do. And it can't be just, well, we'll make regulations, because that's a really difficult thing to do. And politicians like to demagogue on regulations and make sure that, oh, I'll stop all the regulations. Well, not all regulations are bad. And sometimes industry, you know, wants to know exactly what they're supposed to do. So Hank started to write a number of guidelines. And one of the guidelines that he had a big hand in was a guide to inspection of cleaning validation processes, issued in July 1993. And that guideline, still available on the internet, is still an extremely useful document. He's codifying, and by that I don't mean anything legal or regulatory. I just mean he is writing down principles that are still useful today in terms of how should we conduct cleaning validation studies. Hank eventually went on to Johnson & Johnson, and he was there for a number of years, and unfortunately he passed away a number of years ago. But I like to bring this to the forefront that there are you know, human people behind a lot of this stuff, and you know, they make the world a safer place. So what are some highlights that are in that old FDA inspectional guide? One of the highlights is not how to write a cleaning protocol in 24 steps. I just think that I saw that on the internet, how to write a cleaning validation protocol in 24 steps, and I just thought, that's just going to scare the heck out of everybody. I mean, 24, holy smokes, 24 steps, that's just the end. So I put that up there. Hopefully, it might take more than 24 steps, but hopefully you can break it down into little bits, and once you have a good background knowledge of what you're trying to do, you can get your cleaning tasks through. Okay. All he's doing at the time in 1993 was saying that I'm sure they scoured the GMPs at the time, like, where can we, force these people to do cleaning validation studies? And all they could come up with was equipment must be kept in an orderly manner. Now, there was lots of, published studies out there that said, we should be doing cleaning validation. The C in CGMP was, this is a concept that's going to make people safer. But in terms of law, in terms of, US regulation, there was really nothing else other than keep your equipment in an orderly manner. He also noted, by the way, another seminal scandal where a steroid manufacturer had contaminated non-steroidal products with steroids. Steroids, of course, are going to be a lot more active, you would think, in general than other products. So even back then, they're talking about a potent product and a less potent product made in the same equipment. He quotes what back then was probably called a regulatory letter. I think it was just before warning letters. The firm did not consider byproducts and reactants in their validation. So API manufacturers need to take heed, but also just about anybody who's making things with more than one ingredient needs to take heed here. Maybe not with byproducts if you're formulating, but if you're manufacturing a drug substance, you're taking heaven knows how many inputs, 20 inputs, 30 inputs, and you're getting out at the end one nice, clean, pure thing. In formulation, you're taking a few inputs, a couple, 3 excipients, the active ingredient. You mush everything together, and you get one thing out. Everything in the everything in the formulation, maybe there's very little waste. In the API manufacturing, there's a lot of waste. But you got to take those things into account because those things have been in your vessels. What are they talking about? Well, written procedures detailing the cleaning process still needs to be done today. Procedures specific to types of equipment and types of cleaning. Is it a water-soluble substance? Is it an organic soluble substance, etc.? You have to decide how you're going to validate, write a protocol. This is what I shall do. This is what I will validate. And it notes that cleaning between batches of the same product, well, then the standard can be visually clean. And that's more or less, that's probably still true. The one caution I would give everybody there is if you happen to know that your product is unstable, and so you're going to get stability impurities over time, then visually clean in between batches of the same product may not be good enough. Also, I don't think this guideline anticipated that you would always do the crystallization in a vessel. Crystallize, crystallize, crystallize in that vessel. You're not doing anything else in the vessel. So you have to clean in between crystallizations. And the answer for that has almost always been no. I don't have to clean every single time, but I should set some sort of standard when I'm going through a campaign cleaning like that. What kind of expectations did the FDA have way back when? Well, there's a first mention that I could find of dirty hold times, right? Talked about hard to clean areas. Talked about microbiological cleaning, right? If that's important. Validating the analytical method, right? Analytical method validation was a relatively new concept back then. How are you gonna prove that the vessel's clean? Are you gonna swab it? Are you just gonna test rinses? How are you gonna do those things? They noted that the agency was not at that time going to tell you how you set your limits, except there's those words again: practical, achievable, and verifiable. Now, regulatory agencies have been talking about how to set limits more and more lately, and we'll get into that. Notice that our lives are supposed to be, in the United States, around the turn of the century, union organizers would talk about that they wanted the day split into 3: for sleep, for work, and for leisure, 8 hours, 8 hours, and 8 hours. But of course, what we have today is we work a lot, and then we goof off, and then we have crippling anxiety for some of the rest of the time. And let's try not to have so much crippling anxiety about the topic of cleaning validation. So what I'm advocating and what I'm going to talk about here is a phased approach. And in no particular order, we have some phases that we should go through while we're developing a good, tight, defendable cleaning validation program. Notice that the phased approach, the stereotype is that nobody ever gets anything started during a phased approach, or finished rather during a phased approach. And if we're not going to worry about that, we're just not going to work. We're not going to plan. We're just going to get boom. We're going to go. Then what happens? Well, people just don't even start. I watched an experiment once where we were supposed to talk about. I was a timekeeper, and I was supposed to see the group, particular groups, and see how much time did they spend planning during this experiment to do the task. And a guy was working for me was in one of the groups, and he immediately took over the group, and he said, "We're not going to plan. We're just going to go." And they only got half the task done. It was a really interesting. The groups that planned did get the task done, and the guy who was working for me, hopefully I didn't teach him that. He decided we weren't going to plan. So let's try to plan a little bit. So what are we going to do? Well, first of all, we're going to need a lot of data on our products. Okay. We're going to need to set limits, and we're going to need to know how are we doing that. We're going to need the analytical method development team. We're going to need some chemists, some analytical chemists. They're going to need to come in here, and they're going to develop some methods for us, right? We're going to need to validate various things, not just that analytical method, but we're going to need to validate it as part of, can we detect the product at the limits we're going to talk about? So we're going to need the limits. Can we detect the product on our product contact surfaces? The surfaces are going to be important now. It's not just, can I see residue in there? It's not just, is there something in the waste stream that I can worry about? It's going to have to be, did I leave anything behind? And is the method itself valid, quote unquote? Okay. Hopefully we're not going to have to go through method validation in terms of the way you would validate an assay. But hopefully maybe as a limit test, can we validate this method? Then how are we going to clean? A lot of firms don't really think about that. Do you want to have a one-size-fits-all cleaning procedure? Do we want to have a couple of families of cleaning procedures, right? Maybe the products that are very soluble in water, we're going to say, okay, we'll clean those in a certain way. Other products that have terrible water solubility, maybe there's other ways we can clean those. Then, of course, you have to worry if I'm going to use a lot of organic solvent in my cleaning. Yikes, what does that mean for my waste handling? What does that mean for my environmental side? Safety, those kinds of things. Then we're going to gather at some point some data from the laboratory, hopefully, from the plant. You might need some data from the plant. Can we clean at scale? I think cleaning is a lot harder than, and folks who have piloted processes and then scaled them up will tell you, gee, that was really tough. And the pilot helped, but it didn't tell me everything I needed. Can we clean at scale? Scaling up a cleaning is, in my experience, can be quite difficult and worse than scaling up a process. Once we have a lot of data together, then we're going to write a protocol, and we're going to document how we validated our cleaning. Then It used to be that was the end, right? But in the last, no, it hasn't been, it's actually been longer than you think. You want to maintain the validated state, right? Validation isn't just an exercise and we throw that in a pile and hopefully we can find it when the regulatory agency comes by. It is something we should be talking about as a cycle all the time. Validation should be an ongoing thing. Now, maintaining the validated state, I said there's no particular order. Obviously, that has to be last. Oh, wait a minute, maybe rejoicing is last. And I will argue a little bit that you really do need to rejoice when it's all over, because hopefully, if you've done this right, you should be saving your firm time and money. Okay, so the first thing we're doing is gather data. What sort of data do we need? Well, hopefully the Sopranos aren't going to help us gather that data. If Paulie goes to help us gather the data, it's going to be wrong. Actually, none of those guys were. I'm looking at that, and none of the. Maybe Silvio was not so bad, but all these guys said silly things during the show. What are our inputs? What vessels see which inputs? What vessels see which product? What vessels see waste? You'd be surprised how many people will test a vessel for a product, let's say, that they manufacture, and that vessel is used only to stage an excipient, or is used only to stage a reactant during an API process. And then they'll say, look at that. The vessel is 100% clean, 0 ppm product found in there. Well, of course, 0 ppm product found in there. The product was never in there. So we need to know what we're cleaning for. Do byproducts matter? This is mainly an API thing, right? Kind of depends, right? Do impurities matter? Well, if I clean, I get into an impurity. My final substance, the impurities, it's kind of an impurity of an impurity at that point. It's going to be vanishingly small. But you got to think about these things. Do I know anything about the toxicity of these byproducts? Sometimes a byproduct, of course, is going to be a salt or something like that, and you're going to have as much on a molar basis of that as you do of your active ingredient. And then, okay, now I got a real worry. How did I get rid of that byproduct? If it was a salt, what am I doing to make sure that I can use that vessel again? Or is anything of particular toxicity or potency? How about my excipients? Or if I'm doing a formulation, a cream, is there anything there with known skin sensitizers? If I'm going to go to a cream from some other product, what am I doing there? You want to consider solubility of all your inputs and outputs. Okay, it really doesn't help you if you're using, say, activated charcoal in a product, right? And that doesn't dissolve in anything. So what's the solubility of that? Well, 0. I got to worry about that when I'm cleaning out the vessel. You might get some activated charcoal in your next product, and you say, oh, come on, it's not toxic. Put a teaspoon in front of me, and I'll eat it. And that's true, because it's a drug for, it's your grandparents may, if you still have in their medicine cabinet, some activated charcoal that they take when their stomach's bothering them a little bit. It's in the USP. So what? Nope. It'll get your product rejected and it'll get you in trouble because, again, it's a near miss, right? Yeah, okay, there was only activated charcoal left over behind, but wasn't that supposed to do things in terms of purifying your drug, and is there any drug left over, et cetera, et cetera? You're going to need to know your LD50s, if possible, or ED50 or LD low, or any kind of toxicity kind of information that you might find somewhere. The no observable effect limit, if that's known. Genotoxicity, teratogenicity. That's a tough one for me. But that refers to a problem that a developing fetus might have. Those sorts of things. So if that information is available, you're going to need to know that stuff. Dosages. What's the minimum formulated dose? What's the maximum expected daily dose? Does the thing a sensitizer? How about excipients? Are they sensitizers? The byproducts, are they sensitizers? All of a sudden, the byproducts become much more important. Are the raw materials, are they sensitizers? If I only had raw materials in a vessel, I got to worry about that, right? That's what I'm worried about cleaning. I'm not worried about cleaning the product I was trying to make. You're going to want a method that's extremely sensitive, right? But then if you have an extremely sensitive method, from my past, we would clean to a low UV limit in our vessels. So we would test rinses for UV activity. And if the UV activity was really low, that told us a lot about what we had removed during the cleaning process. But we had a naphthyl group on one of our products, and naphthyl groups really have a lot of UV activity. And so it would be like really, oh my gosh, you would figure out, well, what's the limit of detection of this naphthyl group in the UV? And it was super, super low parts per trillion. If I did that, am I going to constantly fail even though I'm cleaning to some ridiculously low limits? You got to think about that too. You need to know your vessel product contact surfaces. How big are they? What area do they have? If you're going to test a rinse, what are the volumes of those rinses? What's the smallest volume that you're going to test? What are we going to swab with? When I first got into this game, these many years ago, there really wasn't swabs that were meant for cleaning, right? So you were going to swab, well, we'll swab with cotton swabs that they're used. Our micro lab uses a bunch of the cotton swabs. They got a big bin of them. We'll use those. And then it would turn out that the cotton swabs had oils in them that interfered with the low levels of detection you need while you're going through cleaning. Nowadays, you can buy things. This is one particular company that sells swabs that you can use for cleaning, and those you can count on. Although do an experiment, you can count on that they're not going to have residues that are going to interfere with your methods. How big an area are we going to swab? Because that becomes important to your limit calculation. Maybe we're not going to swab. No law says you have to swab your vessels. It's expected. You would have to have a heck of a justification if you weren't going to do it. But maybe we're not. Maybe we're just going to look at our vessels and we can seal our contact surfaces, and that will show that they're clean. That used to be an argument that you could make on a sort of a kind of basis. Now it's a tougher argument to make. Ask yourself why I put that question there. From the beginning, the objective of the inspection is to ensure, this is again from older documents, that the limits is scientifically justifiable. So make sure you write a document approved by your quality unit that states how your limits were determined and why they're good limits. Why are they justified? In all cases, you're going to need to calculate a maximum allowed carryover. One does, as Aragorn will tell, or that's Boromir, I guess. One will not just set limits. Are you going to set limit based on the entire train of equipment that you used? If you never break the train into its smaller component parts, you can say, I will release this whole train at the end based on a limit. If you do break it up, if you take the mixer and you put it over here with a different set of vessels, and you take the crystallizer and you put it over there with a different set of vessels, then you're going to need limits based on individual vessels, depending on the flexibility of your plant. Are you going to want to have multiple limits based on the known next product? I worked in a facility where I was looking at the cleaning validation limits, and we didn't have any. And I said, well, why not? And he says, well, we calculate it every time based on what's coming next. Well, if you calculate it every time based on what's coming next, you have an awful lot of validation to do, because you've got to make sure that that limit is validated for, there's going to be a number of limits, hundreds of maybe of limits that you're going to need to have, make sure that you have validated. Are you going to want to validate based on one worst case next product? Well, there you're going to get one limit for everything, but is that going to force you to do way more cleaning than you need to do? And then, of course, the big question, I got to calculate a MACO. What basis am I going to use to calculate the maximum allowed carryover? One of the traditional limits, one I followed most of my career, was for swabs, was a dose-based limit. So you would figure out the dosage of the product being cleaned, figure out what the smallest next batch size is going to be, and then what was the dosage of the next product. And then you would have a safety factor, and that safety factor was usually 1,000. You would want 1/1000th of a dose of product A allowed to carry over into product B. Where did the 1/1000th come from? Well, it was kind of out of the air, kind of. There was this truism that 1/10 of a dose of a product was ineffective. And then we throw in 1/100th, just to throw in 1/100th, just to make sure that you got your limits down, fairly low. You're going to want to calculate the maximum allowed carryover for all possible plant combinations. So there's a spreadsheet in your future, right? If X is going into Y, what does that get me for a limit in my plant? And then you go through all your X's into Y's and try to pick the worst case scenario so that you get one limit for every product in your plant. And I shouldn't say one limit. When I say that, I mean each product has its own limit, okay? But you're not relying on knowing what the next product's going to be in the plant. You're saying, based on a worst case scenario, the limit, and when I'm done with X, product X, and I'm cleaning, I get to this limit in my vessels, I'm done, I can release it. I don't care what's coming next because I know what's coming next is going to be okay. You got to apply this carryover to your vessels. If your vessels are being released individually, or if you're releasing one train of equipment all at once. And by vessel, I mean mixers, dryers, hoses, filters, centrifuges, what have you. You're going to want to make sure that you know that stuff. Your carryover is going to have to be corrected for the surface area of the vessel. So if you take your carryover, divide that by the allow carryover, divide it by the surface area of the vessel, that'll give you a carryover per unit area of your vessel. Then you're going to swab, okay? I'm going to swab 10 centimeters within the vessel. Now I'm going to get a Mako per swab area, okay? Then I'm going to take that swab and more than likely digest it in a solvent. So now I've got an amount of residue that I have on my swab. I put that in a certain amount of solvent, do that calculation. Now I've got a concentration. And I've got a limit that QC will understand, that the lab people will understand. It'll probably be in parts per million or parts per billion. I'm going to say I cannot allow any more carryover in this swab that I'm giving you than parts per million, parts per billion. Oftentimes you go on an inspection, people say, we cleaned to 10 parts per million. That doesn't have any meaning unless you know what your surface area was of your vessels, unless you know how much swabbing that you did, how many swabs, all that sort of thing. Likewise with rinses. You're going to have a carryover limit, right? You're going to do a Mako calculation for the rinse, and then you're going to take the last rinse that you did while you were cleaning the vessel, and you're going to say, okay, I would allow yay many milligrams of carryover. I had yay many liters of final rinse, and so that gives me a limit in a volume in a rinse. And the idea, by the way, behind this is that I'm assuming that what's in my rinse is what I left behind. Likely, you left nothing behind, and what's in your last rinse is a lot more than what you left behind, but you never know about a thing like that. So that's a traditional way. Now, though, what are the regulatory agencies of the world saying? Well, they want toxicity-based limits, and there's an EU guideline, GMP Annex 15, applicable since October 2015, at least the most recent, and it requires cleaning validation to be based on a scientific and risk-based approach. Dosages approach this, right? Dosages you're getting there in terms of a risk-based approach, right? But maybe aren't exactly what the agencies are worried about there. There's more things about a permitted daily exposure limit, an acceptable daily intake limit, an acceptable daily exposure. And those things get more derived from knowing the toxicity of the drug and the potency of the drug. There's a thing that I don't understand all that well called some sort of index, I think, of the product, which is what is the difference, and somebody can tell me eventually, but what is the difference between the dosage and the toxicity? Some drugs, that's a really narrow range, right? The dose is approaching the toxicity. And so those drugs have to be administered very carefully. Other drugs, the toxicity is some enormous value, but it has a really fairly low value for the dosage. So those drugs are, once formulated properly, are actually quite safe, because if there's a tiny mistake in the formulation, it's no big deal because you're way, way below what the toxicity of the substance would be. The FDA has not adopted this formally, but we can expect that they will follow it informally, the C in CGMP. What are the Europeans telling us? The visual check, of course, is still valuable. You have to do a visual check, but there were loads of companies out there, apparently, that were saying, I'm cleaning till this is visually clean. And then when they would get asked, what does visually clean mean? They would say, it means it's visually clean. Well, that doesn't tell you anything, right? Because there's some sort of visual limit of detection of residue on the vessel. If you have scientifically supported your limit as being, I'm cleaning until this is visually clean, you can still do it. You have to scientifically support it. That means you have to have good, extensive studies with more than one person. You don't just go find a guy who's got 20/10 vision and look what you got to find regular people, and you got to do some experiments and some statistical analysis, and you got to say, we could detect this amount of residue in our vessels of this drug. And okay, fine, backed up scientifically, good, you can do that. The EU recognizes that there might be a period where you're at full scale, but you're not ready to validate yet, because you've got to gain a little data at full scale about your cleaning studies. They want you to, and this has always been true, but it's mentioned where you want to assess the variable factors in cleaning. If you're manually cleaning, right, and Hercules comes in one day and he scrubs and he gets the vessel clean in one shot, right? Well, what does that matter when the next day somebody comes in who has missed the gym over the last 60 years, like say me, and isn't very good, isn't going to get as much, push as they're cleaning the vessel. So those factors are there. They now formally ask that bioburden risk be considered. They want clean and dirty hold times considered. And they want to know how many you're going to campaign. Fine. It's the same thing in the vessel over and over again. We get it. We're going to allow that, except justify it. Tell us why it's okay for you to go 5 lots before you do an intra-campaign cleaning. Plants I worked in generally used a very limited number of vessels when we were manufacturing a product. So the same vessel would get used several different ways within a process. So because of that, that vessel had been cleaned 3 times by the time we cleaned it at the end. So we never ran into that as an issue. But other plants that might make one or just a few substances, they've got a dedicated crystallizer, they've got a dedicated centrifuge, they've got all these sorts of things. Okay, let's have some justification there of how long you're going to wait before you go cleaning. And of course, now if we're going to do toxicity, right, we're going to need to modify our limit calculations a little bit. So some sort of toxicity value, a permitted daily exposure, let's say, and then again, the smallest batch, say, of the next product, and then the maximum daily dose in the product that's following, okay? And again, and I think have this here, is perhaps a safety factor. That safety factor would depend on what you're doing here with the permitted daily exposure limit and that sort of thing, okay? Now they want you to know, okay, if I'm taking a drug, how much of that drug am I going to take every day? Again, you need to adjust for surface area, for swab area, for volumes in terms of rinses and things like that. Be sure your tests accounts for detergents and solvents used during cleaning. Make sure that you can tell with a straight face that I have a limit for the detergent that I used while I was cleaning out the vessel. How am I going to detect the detergent? So a lot of them are UV active. So maybe I'm going to look at a simple UV test to make sure I got the detergent out. Most of them are going to be total organic carbon. I can do that. Or if they're salt kind of things, I can do conductivity. I could do pH. And those tests are also quite useful because they'll help you account for byproducts, reagents, and other excipients. You should outline a general approach. How am I approaching these things? You know, what am I worrying about there? Don't just say the drug is the worst thing in there. That's what I'm going to worry about. Have a reason better than that as to why you're going to worry about those things. And document this. When you figure out your limits, document how you're going to do that. Well, if you've been in the industry for a long time, right, and the European Union or somebody important comes up with a new thing to do, it's a change, I better run. Nothing scares the pharma industry more than change. So if your plant has a legacy system, right, and you have these, I did all this work and now we got these new standards, recalculate your limits. It's an exercise, okay? Sit down, gather data, and say, okay, if I was to do this in a more modern way, what would my limits be? And you'd be surprised. Very often, they'll be higher than the limits you did before because the safety factors we took into account way back when were pretty large because we didn't know as much as we know today. So we would say, well, I better throw in a big safety factor because otherwise, this isn't going to go well. Now that you know all this stuff, you know, now that we have a lot of data about a lot of things, you go through, you calculate the limit, your limits are higher. Ooh, yay. That means, again, document. I did this exercise. My limits were higher than the limits I've been using. Limits I've been using, those are already validated. They're not killing me. I'm going to stick with them. Great. You did what the agency wanted you to do, and you're telling them you're doing actually more than they needed you to do. So if you're not going to use that and revalidate, say why. Say, this is why. I'm not going to, I acknowledge that you asked us to take this approach. If we took this approach, we would have stricter limits. Hooray. That means we would have less strict limits, rather. We're not going to do that. We're going to stick with what we've got. If you do come up with stricter limits, though, and you think, I hate this, I really don't want to go down this road. Okay, say why you're not going to go down that road, okay? Don't just say, I'm not going to go down that road, get the shredder. No, say why you're not going to go down that road. Be upfront. Some more toxicology considerations are now out in the literature. So for example, the permitted daily exposure for anything that's possibly genotoxic, teratogenic, et cetera, is 1.5 micrograms per day. That started to get established as a permitted daily exposure for impurities in drug products, right? Now it's, if I get a carryover into a drug product, well, I've made an impurity in that drug product. So I darn well better think about 1.5 micrograms per day. That, if you want to read it, you can. The agencies will tell you how they came up with that. It's the idea is getting one less excess cancer in 100,000 people. This can lead to some very low limits and challenging technology. Fortunately, drugs tend to go in parallel, so you get some limits. We made some really potent painkillers in my previous life, and you would work out the limits. then you would find out that you get these impossibly low limits if I want to reuse these vessels. But then you would also find out that the amount of product you were going to make was really small, and so you say, okay, well, I can buy glassware. We're going to do this in glassware, and we're going to dedicate the glassware. But figure out what those limits are going to be. Some limits wind up very high, and this is where practical comes in. If you can get to a lower limit easily, you got to do that. You can't just say, this stuff is not toxic at all, and neither, it's no big deal if it carries over. You can't do that. If you can get it to get out of your vessels, you got to do that. Now we're going to do some method development, and this has been my experience with laboratories over the years. When the ice cream man comes, you're just not going to get that much work done. Cleaning limits can lead to detecting trace quantities. This is much more around detecting trace quantities. It's much more around the way you develop a method for impurities than it is for developing an assay, right? You're going to have much lower concentrations, and practically speaking, when you go through testing this. So think impurities rather than assay. Think about interferences. Nowadays, the interferences have been minimized. There are swabs that are out there that are meant for cleaning validation. They shouldn't have any interferences. But you're buying gloves, and you buy those by the truckload. So maybe there are going to be some interferences from your gloves. We always used to do a glove blank. We did a swab blank. Why? Because you might find those some stuff from those things in your product, and they can interfere with testing your method. You might need to look at the Tyvek or whatever you're doing when you're doing these tests. Keep running blanks until you understand that, no, I don't have these interferences. Okay, fine. Document that then. I ran a bunch of blanks and I don't have interferences. Do I have to, when I'm developing a method, does that method have to be specific for the drug that I'm looking for? And the answer really kind of is no. You can do a total organic carbon test. You can do a UV test if they're sensitive enough. There are colorimetric tests. As I said, there's visual testing is still allowed. It's hard to justify, but it's still allowed. Interferences in UV, for example, are going to be additive, right? So if you had half of something that should fail and half of something that should pass and half of something that should pass, they get together, they make your UV fail. Gee, if I had more specific methods, I would have passed the vessel. But they can help you to achieve very low limits. But again, the caveat, the reason for the failure may not be obvious if you don't have a specific method. So almost universally, well, I mean, I have seen other methods used, residue on evaporation, all those kinds of things. People are still plating TLC plates. But usually what you see out there is HPLC. UPLC is quite useful for methods because, and those are specific, right? But you're going to need to validate the limit. A limit test should be appropriate as validation, right? But what about those other things that I don't do when I'm validating a limit test, right? Well, those other factors can aid me in a failure investigation, right? If I've got problems with my vessels, they're not passing. Why is it not passing? The more I know about my analytical method, the better off I might be. Now, of course, there's the added dimension during method validation of the recovery study. I'll talk about that in a minute. I'm going to talk about it now, actually. The limit itself, right? The pass-fail calculation has to be adjusted for the recovery from the vessel surface. Remember, you're not just testing a pill. You're testing a rinse or a swab that came from a vessel surface. Maybe the material sticks to the vessel really well. If it does, that means that you might have only got 50% of the stuff onto your swab. So you got to know your surface areas. Common ones in pharmaceuticals: glass-lined steel, stainless steel, 316 stainless, Hastelloy, Teflon, plastic. And you can matrix this, right? You can say, let's try a recovery factor from a worst-case surface, but make sure you're justifying that, right? You might get away with saying things don't stick to Teflon. Make sure your quality unit, of course, is involved in all this review. Your solubility data might help you justify the use of the least soluble product to get to one adjustment limit, one adjustment from your surface swab studies. But what will happen is you'll find out, oh, I'm only recovering 60% from this equipment. You have to take your answer then, and you have to say, I'm going to multiply it by or divide by 60%, and that's going to give me a higher limit. And I better meet that, or I said that backwards. You're going to have to take, I'm only recovering 60% from the vessel. So when I get a swab and I get an answer, I have to take that into account, that represents only 60% of what I left behind. So I have to adjust that number and then compare it to my limit. Another thing that's important in recovery studies is differences in swabbing technique. How do I capture that? A good way is to use 2 swabs maybe every time. I'll swab, put that in a solution, swab again, put that in the same solution. Why? Well, because one guy might be really strong and he gets everything with the first swab. Another guy might not be terribly strong, but he gets everything in 2 swabs. So that helps to get that out and even all that out. Then how are we going to clean? Right? That's a phase that we're going to have to go through. Well, remember, selection of your worst case product for cleaning. If you're going to say, I'm going to work everything based on a couple of products or one product, well, that means I'm going to clean everything the way I would clean that worst case product. So if you're going to do that, right, that's all I'm going to validate is that one worst case product. Now I'm cleaning everything that way. So take that into account, right? You might be way over cleaning. You might have to hive off a product or two and say they get special attention in terms of cleaning. We're going to clean everything else a certain way. These 1 or 2 products we're going to clean in another way. You've added some validation time, but you've subtracted a whole bunch of cleaning time down the road. I would encourage the use of physical cleaning methods, meaning things that are not human dependent. So CIP spray, clean in place balls. Clean in place balls, let's see. Oop, there's a clean in place ball, right? What that does is that cleans the vessel pretty much the same way every time. Takes the human factor out of it. And I think that's an important thing for all of us to do. Reflux is a cheap and easy way. That just means boiling, right? It's a cheap and easy way to take the human factor out when you're cleaning a vessel. If you fill the vessel and you agitate it, right, that takes the human factor out of it to some degree. Although there, of course, you could be using a lot more cleaning agent and generating a lot more waste than you want to do. And so all these things either create or lower the waste stream that you're going to have to deal with at the end. You can come up with a wonderful cleaning validation procedure and you think, oh boy, the boss is going to love me. And it turns out he came out of environmental health and safety and he sees the bills for getting rid of all the waste that you generate, and he wants to hit you over the head because you now are generating a lot of waste while you're cleaning. And that's not good for the people either. Again, more about how we were cleaning. I said manual cleaning presents real challenges. You're going to get variance from person to person. Valves in a vessel. Valves hide product. They all have their pluses and minuses. The butterfly valves and all these other valves. Product will hang behind those. And what do I do about those, right? Try to make sure that those are included in all of your cleaning. Try to make sure that you're pushing things through all of that stuff. If you have hard pipe connections, am I holding up product? Is that product in the walls, where the piping is, and I can't get to it? As you're going through cleaning validation, consider an endoscope. Consider looking through those piped connections to make sure that you are, in fact, getting to everything. Placebo batches. People used to run placebo batches and say, I'll just run a placebo batch in between, and now I'm okay for the next product. Nope, that's no good unless it's unless it's extensively justified in a way that I've never seen. But that doesn't mean you don't do it. If you're doing placebo batches, that doesn't mean you don't do them, right? That can be part of your cleaning. It just has to be, it can't be the only thing you do in cleaning. Making your worst case decision. Now we got to sit down. What are we going to consider? Solubility, potency. What type of compound am I using? Is it a steroid? Is it an opiate? Is it a sensitizer of some sort? Antibiotics hopefully are being made in a plant all unto itself. Can I make a steroid in a multi-use plant? I still don't know the answer to that. Consider 2 or 3 validation or cleaning methods. In other words, consider in your plant, maybe not one size fits all. Maybe I want to have a water soluble cleaning. I want to have an organic soluble cleaning. Maybe I want to have a highly potent cleaning versus a lesser potent cleaning. That means I'm doing more than one validation study as I go through, but it means maybe that I'm saving at the other end in terms of most of the time when I clean, I can use this process that's pretty good and doesn't generate a lot of waste. And then there's another time when I make this product that's a real nasty pain, and it always hides behind the valves. And there I'm going to still validate, but I'm going to do that in a way that's separate from the first way I was doing things. Cytotoxics. Maybe I should be rearranging my plant. Maybe I need to make all my cytotoxics in one train. You can think about, in terms of worst case analysis, that one cytotoxic contaminating another cytotoxic is a little different than a cytotoxic contaminating a cough medicine, right? It's the two things are completely different in terms of how scared you should be if you're doing those kind of things. Think about when I'm transitioning from cytotoxic materials to non-cytotoxic materials, or from sensitizers to non-sensitizers, et cetera. What am I doing there? And then document those decisions. Justify everything that you're making in terms of that stuff. Now you're going to want to clean in your plant in the end. This is hard to pilot, if you ask me. And agencies recognize this. So you get on we're going to have a test to clean phase. But the test to clean phase, the days of, we're going to test to clean for a little while, and it turns out 25 years later, you're still testing to clean, that's not going to fly anymore. Clean in place technology is great because you can test it, right? You can do a riboflavin study, for example. You can coat your vessel with something like riboflavin that has a real it's very, very sensitive in fluorescence or in some other kind of test. And then you can use your clean in place, and you can go through it, and you can say, did I clean everything out with my clean in place technology? And if that's the case, that gives you a really good feeling that this technology is reaching every part of the vessel that you need to reach. If you have good data and you have good stuff in the back, you're going to have a good protocol. Okay. Now, clean and dirty hold times. For existing products, you can data mine to get this information. What was the longest we ever held these vessels before we started cleaning them? There, and was that successful? If that's the case, there you go. There's a dirty hold time. How about for cleaning hold times? Well, for clean hold times, what am I doing with the vessel when I'm done? Am I putting it under a nitrogen blanket and holding it that way? Am I holding it under vacuum? Am I in a clean room with that vessel? Am I I'm putting it in such a way that it is not getting exposed to anything else that's going on and around in the plant? You can make a clean hold time justification there, and you can probably do a clean hold time for quite a number of days. If you in the old days, they would have vessels and they would just sort of sit out and maybe they get dust in them. Okay, there you're going to have a pretty short clean hold time, right? Because you think, oh darn, this is going to get dusty. So there, in that case, of course, you're going to have a shorter clean hold time. The dirty hold times, try to use your data to get you to a good dirty hold time. Always visually inspect though before starting. Okay, weird stuff happens, and you look in your vessel and you go, this was clean 2 days ago. How did that get in there? And then you find out that there's something in your piping diagram that you didn't know about. and something backed up into something that you never thought could happen in the past. It's often a very good practice to rinse your vessels before starting, and that's an okay thing to do. Make sure you check though for something like particulates if you're going to do that, because you want to make sure that nothing untoward has happened while you've held your equipment. And like I said, how'd that get in there, right? Well, how'd that get in there? You find out that the piping diagrams are not as good as you thought they would be. Then you're going to write a protocol. Consider a master validation plan, justification, limits analysis, what kind of matrixing strategy. Make sure that this is where you document everything. How are you cleaning? What tests are being done? When will you revalidate? Et cetera, et cetera. Put all that in your protocol. Finally, what if you introduce a new product? Well, you should have a system for that. Every time a new product is introduced, you've endangered your worst case assumptions. You've now said this new product's coming in. So you got to check. We're going to make this new product in here. Do all my assumptions still hold? If they don't, you've got change control, and you're going to have to do something and maybe go back and start from the beginning. If not, you can document all that and say, nah, this guy's really easy to clean. We're putting it in the plant. Everything still holds. Maintain your validated state. And change control here is the key. Ensure cleaning is considered in your change control, right? You got a new product, you got new equipment. What does that mean for your validation? If the change has had no effect, you got to state that. I can't tell you the number of firms that get into trouble knowing that everything is fine, but not having it written down. If it isn't documented, it isn't done. Evaluate the state of your cleaning validation. A good place to do this is in your periodic product reviews. That tells the agency that, yes, you are thinking about all this kind of stuff. And finally, at the end, rejoice, because if it's done properly, cleaning validation should save you money, right? It should keep you out of investigations. You should be able to reduce your solvent costs. You can automate a lot of systems, maybe. And that winds up, that in the end should save money and should save you time and effort down the line. Build the system. This is from, I learned this from painful experience. Build the system so that it runs itself. Intervention by specialists and management, of course, raises costs. If you have to have, there's Fred down the hall, he knows about cleaning. Well, if only Fred down the hall knows about cleaning, you got problems because of what happens when Fred leaves or Fred goes on vacation. I want to thank everybody for their time and effort. If you want to contact me, you can do that, and the best thing to do is to contact Brian Coleman, who coordinates a lot of the work that I do at this email address. And we still have some time. I think we had 80 minutes, right? So what's next in terms of questions?
Sounds great. Thank you so much. And we have a couple questions coming in.
Good.
The first one, is it common for plants to consider cleaning procedures based on dosage forms, or does it still revert back to PDE Null type levels to consider? So
more and more the agencies want you to go with toxicity. Where dosage forms start to matter is you can use those as a way to sort of matrix your cleaning. So if I have a dosage form that's a cream, I need to do certain things, right, to clean out that cream at the end. Whereas if I have an oral solid dosage form, I'm going to use different techniques. So there, yes, sit down and say, in this same equipment, I'm going to do different kinds of formulations, and I want to make sure that I have different techniques for those. But the agencies more and more want toxicity taken into account when you're setting limits.
Thank you for that. Next question, do I really have to worry about excipients or byproducts, raw materials, etc.?
I'll confess that I actually wrote this question. And the answer is yes, you do, because they're there, right? And, if you've got a waste stream in a vessel, and that's all you've had in the vessel is the waste stream, you got to know what's in that waste stream. And if you're going to want to use that vessel, I mean, if all you do with that vessel is use it for waste streams and there's no way it can back up into anything else and et cetera, okay, fine, maybe you don't worry about it too much. But then your safety people will say, hey, hold on there. You left something behind. How do I know I'm not going to get some sort of terrible reaction if I mix two waste streams together? So in all those cases, you got to worry about everything that's going in there. That doesn't mean you're necessarily testing for everything that goes through your process, but you're going to want to account for that. Okay.
Great, thank you. And yes, that's true. We did have a couple post questions so that we would allow time for people to send their questions in. So please do, if you have any questions, type them into the questions box. Our second question, Joe thought he might reasonably be expected to ask is, can I rely on the fact that a drug is used for a short period of time to get around the 1.5 mcgs a day carryover limit? Yeah,
so the answer to that is probably not, because the origin of this is when the FDA started to talk about limits for cytotoxic compounds and potentially cytotoxic compounds in impurities in drugs. They came up with this sort of thing. Well, if the drug's used every day, it should be 1.5 micrograms per day. But if the drug is only used for a month, it could be this higher value. And in order to get firms, because it was getting a little tough to make certain drugs. I don't think that's going to fly for cleaning, because you really don't absolutely know in terms of what's being used next and what you're contaminating. Is that used? Is that going to be a drug that people take every day? So I think if you're going to go down that road, go ahead and justify what you're doing. But I don't. If you don't have a written justification, you're going to lose that argument.
Great. We have time for one more question. This one is from our audience. And how frequently should you revalidate and run verification?
So there's no absolute rule on that. What you should do is there is an absolute rule that you really ought to be preparing at least annually a periodic product review. And that's an ideal place for you to evaluate the cleaning and say, what changes have accreted, over time? What's different now? And if the answer to that is, well, gee whiz, this is an aspirin factory, and we've been making aspirin since Caesar was a private first class, then we're doing it exactly the same way, et cetera, you might not have to revalidate very often. But if new things are coming in and there's change and there's a lot of improvement going on, then you're going to have to make that evaluation and do it a lot more often. Right now, the biggest thing that I see out there is people don't do this at all. They don't do the exercise. Firms don't say, my cleaning is still good. So it's easy for regulators to come in and say, well, you're not considering this, and you should be considering that. And that gets you into trouble. But make sure that whatever you're doing, you're documenting it and you're saying, I considered it for these reasons, we're still good. If you do that, then, and your reasons are good reasons, you should be okay.
All right, perfect. Joe, thank you so much for such a good presentation. And thanks everyone for joining us today. This was recorded, so we'll post it on the website so that you will be able to revisit any of the points that you would like to revisit. As mentioned, Joe will provide some resources that are hyperlinked, so we'll be able to give you some additional details. And of course, as always, if the EAS and Joe specifically can be of help to you with any of your GMP needs, please always do reach out. We would love to work with you. Joe, thanks again. Have a great afternoon, everyone.
Yeah, thank you, everyone, for your kind attention.